Hello again, welcome back. Our next 50 minutes, during the next 50 minutes, we will be listening to our final three pitches this afternoon. And after that, we will go into a network session where we will leave the jury to do their important job of deliberating over the 14 pitches of today. We will also have a break later on as well, so that we will be coming back together at 16:45 CET, Central European Time, for our expert insight, followed by the ceremony awards. But now, to kick off, we're going to go to the UK, and we are meeting Agnieszka Janezek, excuse me, from Renovos Biologics. I hope I pronounced that well. So Agnieszka, Renovos Biologics UK, the floor is yours.
Thank you. So, hello, I'm Agnieszka Janeczek, the CEO of Renovos, a company pioneering synthetic nanoclay gels for tissue regeneration. So, regenerative medicine is increasingly significant for tissue repair for both aging populations, but also active younger demographics damaging their tissues through sports. Stem cells within our bodies can regenerate tissues and are triggered by powerful biologics. BMP2 is most widely used and represents a $1 billion market, but current clinical product led to adverse events such as bone growing outside of the skeleton, tissue swelling, leading to difficulty breathing, and regulatory removal from some usages. Yet in many cases, BMP2 remains the only reliable option for promoting bone tissue formation. So to unlock the potential of stem cells and circumvent the safety issues, we need to deliver biologics such as BMP2 in a safe and precise way. So our Renovite nanoclay gels solve these issues to unlock the potential of regenerative medicine. Renovite is a de-risk technology based on over a decade of research at University of Southampton, with validated proof of concept, harnessing the power of BMP2 protein and addressing that $1 billion global market with use in spinal fusion. Renovite delivers therapeutic agents and retains them within the target site of repair. Unlike other technologies, there's no release from the material until patient cells invade it, break it down, in time replacing it with regenerated tissue. It templates new bone tissue formation, as you can see, in the exact space delivered through a precise injection. And secondly, thanks to that retention, we can dramatically reduce the dose, lower the cost of goods, and improve the safety for BMP2 and other potent biologics. Nanoclays are industrial ingredients, and we have refined and validated our formulations, Renovite gels, for therapeutic use. Our nanoclay is synthetic and easily scalable. We have 2 granted patents, complemented by trade secrets and extensive know-how. This first product offers many advantages over competition and fits within the care pathway. External market access interviews with payers and end users confirm there's a clear appetite for a new, improved and safer product and a willingness to pay 15 to 40% price premium. Reduction in the dosages leads to lower cost of goods, so enables entry into more price-sensitive orthopedic segments. This provides more economic solutions for healthcare systems and better outcomes for patients. So the nanoclay platform is a breakthrough technology that can ultimately revolutionize regenerative medicine. Our R&D program also includes point-of-view cell delivery matrix, as well as wound healing and antimicrobials, allowing Renovos to grow and scale up, addressing the areas of largest unmet needs in the wider $5.7 billion orthobiologics market. Our pipeline and our mission is to help clinicians deliver safer, more efficacious regenerative medicine treatments for tissue repair and enable that wider regenerative medicine ecosystem. We have an experienced team from world-class academics through hands-on clinicians to industry veterans who have built startups and operated at corporate level, commercializing orthopedic innovations in the spine and securing successful exits. Renovos has been endorsed by several organizations, including by Orthopedic Research UK, a charity, by making their first ever equity investment. So our business model relies on internally generated assets, driving both our partnering and licensing strategy. We have built deep expertise in the use of Renovite, its manufacturing and formulations, and de-risked the technology with our preclinical studies. We are aligning further development with industry expectations and are in discussions under NDA with major players. We are now raising 10 million in our Series A to reach value inflection points and enable human trials. With this funding, we'll establish GMP manufacture, generate GLP data required for partners or licensees for the first product, and we have also multiple exit options, all yielding generous investor returns. This funding will also allow us to scale up the rest of the portfolio and continue developing other innovative solutions based on the Renovite platform to create further value and address the biggest healthcare needs in the growing regenerative medicine sector. So that's your opportunity to invest in the future that we're building. So if you'd like to support our vision, join us and get in touch. Thank you.
Thank you very much indeed, Agnieszka. Thank you for your very, very good pitch. So let's now listen to our jury. Here they are, to see who would like to ask questions. And Renato, I believe you have a question for Agnieszka.
Thank you very much. My question is about the point you raised around the payer interviews. I would like to know a bit further what kind of payers in which markets and what's their willingness to pay for this new therapy. Thank you.
Sure. Thank you, Renato, for the question. So we have conducted external market research, and that was actually funded under Innovate UK. We're able to outsource the market research, and we were able to interview end users, so key opinion leaders, clinicians, and also payers in focusing on the US market, Germany and France, in Europe. So we had a small cohort of interviewees there, and that price range premium that I mentioned is the price premium over the current Medtronic's product. They indicated a 15% price premium. That was actually from German feedback. So normally quite conservative. If we offer a safer product in comparison to the Medtronic product, even up to 40% price premium in the US, if we are also able to increase efficacy versus Medtronic's product.
Alan, please go ahead.
Thank you for the presentation. I mean, I guess given the history with the MP2, the regulators will be, I guess, super sensitive to any potential side effects. So as you look forward towards the clinical development pathway and safety data, do you think there will be a high burden in terms of longer term follow-up outcome measures that will significantly increase the cost of getting the product to market?
So the short answer, yes, the regulators are obviously more sensitive in this area, but actually any new spinal applications, spinal biologics now require 2-year follow-up. So obviously in the initial study endpoints after 12 months, but actually you would want to have that 24-month follow-up as well. And yes, safety aspects are the key differentiator for us and that key retention in our nanoclade technology. So we will be designing these trials accordingly to get the maximum data. But yeah, 24 month would be the requirement.
Yes, Iris, go ahead. And Emma also has a question. Please go.
Yeah, it is a short question whether you have already identified any partner for the preclinical and the clinical development.
Yes, so because spinal fusion and models are more well known in the US, there's various CROs that operate in that space and run those routinely. So we've been talking to the few usual larger suspects such as Charles River, for example, but actually identified also smaller non-for-profit CROs that have very good experience in that space and align with what FDA would like to see in those models and further development. So yes, we've got various quotations and all that path planned out right now. So we're just looking for the correct funding to really kick off that program.
Thank you. Emma, I believe you would like to ask.
Yeah, very fast. Of course, safety is the key issue here in relation to the product. But then when comparing efficacy, what are you looking for? Is that the time of recovery, posology regime? Can you comment a bit more on that?
Yes, so with time to recovery, that's now our key focus really. It would be more then no need for revision surgery. For example, in spinal fusion, there's quite a lot that fail and they need additional spinal fusion following that and revision surgery. So we want to ensure that the first attempt is efficacious enough to try and circumvent that. With regards to, like you said, the main feature would be the safety aspects and lack of those adverse events that were experienced by patients with the other products.
We have just under 1 minute. Would anybody like to go ahead and ask another question? Iris? Okay, for you.
Yeah, might be a silly question, but you mentioned that the formulation has the capability of reduce dosing, but also improve how you retain biologics. And my question is, are you thinking, or have you tested it with any specific drug, or is it regenerative?
Yeah, so we've been working in small molecules, protein, also cells. So it really is a platform that's easily tunable to various types of differently charged molecules and sized molecules. So we have loads of data supporting other applications beyond BMP2 as well.
Thank you very much indeed. Well, that completes our very rich discussion between Agnieszka and our jury. Thank you, everyone. Let's now let's go to Spain, or in fact, Boston, where we're going to meet Beatrice Ramuzzi at Arthex Biotech. Hello, Beatrice. Lovely to have you with us this afternoon. And the floor is yours for 5 minutes of pitch.
Hello, thank you very much. Hi, my name is Beatrice Ramuzzi. I'm the CEO of Arthex Biotech. And let me start by introducing you Balix family. Their lives are severely affected by myotonic dystrophy disease. This is a rare genetic, very complex disease which is causing cognitive dysfunction and muscle wasting, mainly due to the loss of function of the proteins MBNL. Although the genetic cause and the mechanism of the disease is very well known, there is still no treatment for myotonic dystrophy for several reasons. First, DMPK, which was the conventional target, the gene where the mutation was located, has been proven to be a challenging target because previous clinical trials focused on it has already failed. It has a complex mechanism of action with a myriad of molecular effects downstream that cannot be addressed just focusing on one single gene. And the main tissues affected, the muscle and the nervous system, are difficult to target without causing toxicity in other tissues. So to cure this disease or to treat it, we need to find new targets. And also, it will be better to use a multi-target approach, and we need to improve the tissue-specific delivery techniques. In our case, we have developed ATX01, that is an antisense oligonucleotide against a microRNA, or an anti-MIR, that it's linked to a fatty acid, that it's importantly improving the delivery into the muscle of this molecule. So ATX01 will become a disease-modifying therapy able to address the cause of myotonic dystrophy in a specific, effective, and safe way. How we will do it? We have found a new target for this disease, which is a microRNA that is involved in the mechanism of the disease. It's upregulated in the patient muscles. Our molecule has a first-in-class mechanism of action, and we have improved the delivery into the muscles using this combination with the fatty acid that allow us to use very low therapeutic doses. And why do we believe that we could really help the patients? Well, we have first obtained a robust proof of concept of our therapeutic approach in mouse models of the disease. This information is already published in peer-reviewed journals, and we have validated with the FDA our regulatory roadmap. In addition, we have a strong IP protection with 2 patent families, one of them already granted in US, Japan, and Australia. And we are in a growing market in a very dynamic sector because also, although myotonic dystrophy is considered a rare disease, it is still the most common muscle atrophy in the adults. There is almost 1 million patients all over the world. And we are in the field of RNA therapeutics that right now it's booming with thanks to the successful approval of drugs such as the COVID or vaccine or inclisiran against cholesterol. So this market is really having a huge annual growth. And there is a tendency to see very early deals between biotechs and big pharmas interested in the field. In our competitors' landscape, I should tell you that there is different approaches to treat myotonic dystrophy, but Artex Biotech will be in the part of antisense therapies. And our main differential factor is the fact that we are using a new target and also a new specific tissue delivery technique that allow us to use very low doses in comparison to other approaches. In just 3 years, we are moving ATX01 from our benches in the laboratory of Valencia to the bed of the patients. We are now starting the regulatory preclinical studies, and we are planning to initiate the phase 1/2A at the end of 2022. That will be followed by a phase 2/3 that might be registrational. We have already raised enough to complete all the preclinical studies, mainly between two venture capitals, In Vivo Capital and Advent France, but we also obtained some non-equity funding from different grants, including EIT Health Support, in different programs, and we are seeking for 20 million to complete in the next round the phase 1/2A and also the chronic preclinical studies that will enable future studies. This is our team, the people fully dedicated to the project that will make it a reality. We put together here more than 50 years of experience in different fields, and we are continuously growing, keeping the team lean but effective. We were successful in involving in our project the main key opinion leader in the field of myotonic dystrophy clinical management and RNA therapeutics, and we are very close to the patients because we really need to understand their requirement, their needs. So we are Artex Biotech, a Spanish company that is changing the rules of antisense RNA therapeutics. Thank you very much for your attention.
Thank you very much, Beatrice. Right on time. Okay, well done. Very nice pitch indeed. Let's bring in our jury panel, who I'm pretty sure will have questions to ask. Julien, and then over to Cillian, and then to Emma. Julian,
Okay,
Cillian, Emma.
This type of disease usually come with different onsets and are more or less aggressive or different forms. On which form/onset will you focus for your clinical development? Can you comment on that?
Yeah, we will focus on adults. As you know, it's the disease onset can appear at different stages of life, but we will focus on the adult, what is considered the adult onset above 40 years old.
And what kind of improvement? Sorry, and what kind of improvement do you expect? Slowing down the progression, reversion?
Yeah, in our clinical trial, we will start with a short study of just 6 months. So in these studies, we will perform biopsies in the patients to assess the molecular alterations, just to really see if we see a target engagement. So we will be able to measure the modification of the targets of the microRNA. But in addition, we will measure a panel of splicings that are correlating very well with the strength in the patients. So it will be very difficult to see a real functional improvement in the patients in that short time, but at least we will see a modification of our molecular targets that will be enough to proceed into the next study in a phase 2/3 that might be at least 1 year of duration, because it's a slow progressive disease.
Yes, Cillian, go ahead.
Thank you. I guess a key challenge in the field of oligonucleotide therapies has been delivery into muscle cells and brain
Absolutely.
cells. Interested to know how you plan to approach this, and also which route of administration you're considering.
Yeah, we are using intravenous delivery, and we are combining our oligonucleotide to a fatty acid that we found that it's very, very downregulated in the patient muscles, at least in the biopsies that we perform a study. The fact that the fatty acids improve the delivery of oligos into the muscles was already known, but in this case, in the patients of DM, this specific fatty acid has worked especially well. So in our case, what we observed is that there is a redistribution of the oligo to the muscles, So you see a strongest effect and higher concentration of the oligo in the muscle instead of other tissues such as the liver or the kidney, which will be the preferred targets otherwise.
And did conjugating this fatty acid also improve neuronal uptake?
Yeah, we saw effects in heart, which is also difficult to target, and also in the brain. But we are studying specifically if it can go through the blood-brain barrier and what's the mechanism, because this is just very preliminary data.
Thank you.
Thank you very much. Let's move over to Emma.
Yeah. Following with the Cillian question in relation to the delivery, and then how sustainable is the effect in your in vivo models, and how often you need to re-administer, or what's the plan on this?
Mm-hmm. Yeah, we have observed, that of course with higher doses we have longer effects of the treatment, but at least in our efficacy studies in the mice, what we have done, it's one dose every 30 days, and then we see a maintenance of the effect. We are now going to longer time points because at least with 30 and 45 days, we still see a maintenance of the effect.
Well, I mean, I guess it's a very competitive space as a rare disease. Perhaps you could just briefly recap in terms of what you see are the key features of the target versus some of the other competitors, including some gene therapy approaches.
Yes, the main our main differential factor is our target, because this microRNA is located in the cytoplasm, and most of the other companies that are using antisense oligonucleotides, they are targeting the MPK, that is the toxic RNA that is caused by the mutation. And in this case, this toxic RNA is located inside the nuclei, so they need to go inside each of the nuclei of all the nuclei that it's forming a fever to have an efficacy. So that means that they really need a lot of or a very efficacious molecule, or a lot of concentration of the molecule in the muscles to see an effect.
Thank you, Beatrice. Thank you so much. Ladies and gentlemen, we're right out of time. Thank you indeed. Now, we are going back to the UK for our final pitch number 14 of our Biotech semi-finals. Let's welcome Lily Buzulmat from the company Solas Cure. Lily, the floor is yours.
Hi, thank you so much. I'm Lily Buzelmat, the Chief Development Officer of Solascio, and I will be presenting the work we're doing, which is revolutionizing wound care through biomimicry and evidence-based medicine. So chronic wounds are a huge unmet medical need that we often forget. These wounds are absolutely terrible for the patients. They're very painful. They prevent them from having a normal life. And these wounds can last for years because there's really no good solution. In order to manage these wounds, the typical healthcare system can spend up to 5% of their expenditure to get to this point. Just looking at the US and Europe, there's about 6 million of these wounds, and the problem is only getting worse because these wounds are linked to aging and diabetes. Maggots really are the best option for these patients. These parasites have evolved through millenniums to selectively only eat dead tissue that you could see on these terrible wounds. So they really have great efficacy and safety. However, they're used as a last resort because of the huge disadvantages that they represent. First, it's obviously really terrible for patients to have to accept these wounds on their wounds, but also logistically it's quite complex. So only the most expert wound centers use them today. So wouldn't it be great if we could do maggots without maggots? And that's what we do at Solascure. We have studied maggots and identified the main enzyme responsible for this great efficacy in their saliva. We've cloned that and combined it with a hydrogel. We've got patents on, oh, sorry, I think the clicker was, yes. We've got patents on the enzyme and its use in medical settings and the efficient GMP manufacturing process. And we have complete freedom to operate. This market, as I mentioned, is really big. So even a small market penetration can generate billions of dollars in revenue. We are projecting peak sales around 600 million and 1 billion in a really conservative fashion, just looking at a few markets, and by 2030. Auré's wound gel is positioned as superior to competition across all key criteria. As I mentioned, there's really no good solution, and Auré's with maggots is really the only option that has good efficacy and safety. But we're also superior on other very important criteria, such as psychological impact, pain. Pain is a huge issue with enzymes, patient care cost, logistics, patient burden, healthcare provider ease of use. All of these things we are superior. We have raised 18 million euros to date, and we have delivered against our operational plans successfully. And so now we are in phase 2A clinical studies. We had some key wins with regulators. We've convinced them that we could skip the typical phase 1 healthy volunteer study. And we presented a really comprehensive, successful toxicology and preclinical program, which led them to, for both the MHRA and the FDA, to approve our clinical trial in first in patient. We will have data by 2022, which will be a point of huge inflection, and then we'll be doing our phase 2B studies right after that. Right now, we are raising 12 million euros to fully execute on the phase 2A I mentioned that we're currently in. We'll also use these funds to do our phase 2B randomized double-blind to get data that we can use to start preparing our phase 3 program. And we will also continue to always see what we can tweak about the formulation to make a product that is really easy to use for healthcare providers. So this really represents a unique opportunity for a very fast return on investment in pharma. Looking at market comparables, we are looking at a major inflection point next year with about up to 5 times the current valuation. And at the end of the phase 2B in 2024, we could look at a potential exit with at least 10 times the value that we have now. We are currently in discussions with key wound care players about these exits. I hope this goes. Okay, so this plan is delivered by a world-class team with support from the wound care community, from the industry, and from the healthcare sector. Nobody is learning on the job. Everybody has done that in pharma. There are multiple healthcare entrepreneurs in the team, and we have great support from our strategic advisors directly from the industry. So in summary, this really represents a de-risked commercial opportunity to generate a large return over a short period of time. We have all of our preclinical data. We are enrolling patients. We are in discussions with regulators, and we're looking at an imminent value inflection next year. Thank you so much for your attention.
Very, very interesting pitch. Thank you. Let's bring in our panel, because I'm sure the jury members have questions. Lena already has a question to ask. Please go ahead, Lena.
Concerning the difference from maggots to your enzyme, I mean, from what I understand, the maggots are really eating and taking off the necrotic cells, which probably the enzyme cannot do. So what is the mechanism of action of the enzyme?
No, it's actually not how this parasite works. They have an external digestion system. So what they do is that they secrete a cocktail of enzymes, and this enzyme is a protease that specifically targets fibrin. So the maggot will basically exudate this enzyme, and the digestion of the fibrin, that is the major component of dead tissue, will be liquefied, and then the maggots. It slurps back the digested liquid and then feeds on that. So all of the digestion and the breaking down of the dead tissue is done externally by this enzyme. It also, maggots exudate also growth factors and other things, but really the protease that really targets fibrin is the one that we've cloned.
And concerning the stability of the enzyme, as some enzymes are not very stable, how long do you think, and how do you have to store the cream and all these things?
This is a very good question, and this is why we really insist. We were really happy to have the patent on the GMP production. Producing a protease is extremely complex because a protease will eat itself in the production process if it doesn't have something to eat, something else. So we have actually developed a specific way of producing the enzyme along with something to stabilize it, so that it can be really stable on the shelf for at least a year. Now, we are also, obviously, as I mentioned, we're still trying to tweak it to extend the shelf life to longer. However, in any event, we know that once the enzyme is mixed with the gel, that's when it really activates the enzyme, and it can stay stabilized, active. It can stay active in the patient wound for a long time, long enough for the patient's dressing to needing to be changed.
Renato, over to you.
My question is about fundraising. If I correctly understood, the 18 million raised, 12 to be raised. So what's the strategy, and do you have an ultimate goal to go public or to continue to stay private?
It's again a good question. The way we have developed the strategy is to keep those open. Really, at this point, there really are a few key wound care players who really want this product, because most key wound care players have a hole in their pipeline for specifically this kind of debridement product. The way we are looking at this is that we want to raise these 12 million to do the phase 2B ourselves, potentially with a partnership with one of these key wound care players. However, we will be looking at the potential exit plan that they give us, and if it's really beneficial and good for the investors and the shareholders, then we could consider that. We're looking at either complete exit or partnering. It really depends on what they want, but we have developed the know-how to do it ourselves, and that's why we want to raise the fund, so we have this flexibility to choose the best deal possible.
Iris and Alan, we've got over just a minute, so let's have
Alan,
questions answered. Go ahead. We have time.
Go ahead, Iris.
We have time.
It was just a very short question about the price of the cream. What's the pricing, and whether you need any kind of reimbursement or anything like that for market access?
It's a very good question. So there are discussions ongoing already with reimbursement experts. In the US, there's already an enzyme on the market that all healthcare providers we talk to complain about the efficacy. But the great thing about it is that he has actually carved a path for a reimbursement pathway with Medicare and Medicaid. So we can just use the same code. And if we can show better efficacy, this product has been grandfathered in, and so they have no clinical data. It was put in the market in the 60s when you didn't need to do efficacy trials. So if we show we have better efficacy, we can actually be a priority project on all of the Medicare and Medicaid rosters.
Alan, we have 5 seconds. Would you like to go for it?
No, it's fine.
Thank
you for the presentation.
Our time is up. Thank you very much, jury. Thank you very much, Lily. And amazingly, that brings us already to the end of the afternoon. And that was a pretty amazing conversation around maggots. I think we would all agree. So, to the startups, we wish to send out a huge thank you for the amazing pitches, all your hard work throughout the program, and of course what you are doing to improve and advance healthcare. Equally to the jury, thank you so much also from all of us for the challenging questions, your time, your commitment, the probing questions, and of course your constructive comments. We are going to be shortly taking our final break for networking, conversations, connections, whatever. And of course, we have a very important timeline, because at 5:15, we will be listening to our third and final expert insight. And that, of course, will be followed at 5 o'clock Central European Time by the awards ceremony. We are going to discover who the lucky ones are to go through to the finals. So we are meeting at 16:45. Be there. The excitement of the day is reaching its climax for 5 o'clock. So we see you, all of you, then. Thank you. See you later.